In a cave above the Neander Valley in 1856, a German quarry worker named Johann Karl Fuhlrott held a skull cap in his hands and felt the weight of something he could not name. The bone was thick, dense, the brow ridge jutted forward like a shelf. He'd seen human skulls before. He knew the shape of them. This was not quite that. What he carried out of that limestone cave weighed less than 2 pounds, but the question it asked would take 150 years to answer. And the answer, when it finally came, demolished nearly everything scientists thought they knew. For more than a century, the story of Neanderthals was written in stone, in bone, in the sediment layers of European caves, and what that evidence said was damning. These were not our ancestors, they were our predecessors, an evolutionary dead end, a failed experiment by nature. The textbooks were unambiguous. Homo neanderthalensis had walked this earth for 400,000 years, dominated Ice Age Europe and then vanished, outcompeted by anatomically modern humans who swept in from Africa around 45,000 years ago and simply replaced them. Replaced. That was the word. Clean, final, conclusive. The established narrative held firm for generations. Physical archaeology told a coherent story. Neanderthal sites clustered through Europe and Western Asia. In the caves of Shanidar in modern-day Iraq, where a burial containing flower pollen suggested these beings might have mourned their dead. At Krapina in Croatia, where more than 900 skeletal fragments from at least 75 individuals had been uncovered. At La Chapelle-aux-Saints in France, where a stooped, arthritic skeleton became the template for the brutish caveman image that endured for decades. Their stone tools, the Mousterian industry, showed a certain sophistication, but the consensus was clear. They were cognitively simpler than us, socially less complex, ultimately unsuited to survive in a world that had changed around them. Their lineage ended. Full stop. We modern humans, Homo sapiens, had arrived from a separate African branch of the family tree, passed through them or around them like water around stone, and continued forward. No mixing, no merging, no exchange of genes. The two species had simply inhabited the same continent for a few thousand years of overlap, and then the Neanderthals were gone. It was a tidy explanation. Logical. Supported by skeletal morphology and site distribution and comparative anatomy. Scholars agreed, textbooks said so, museum dioramas showed it, a knuckle-dragging figure on the left, a slender, modern human on the right. An evolutionary gap between them wide enough to drive centuries of certainty through. Then in 2010, a genetics laboratory in Leipzig quietly dismantled the whole thing. The team was led by Svante Pääbo, a Swedish geneticist at the Max Planck Institute for Evolutionary Anthropology, a man who had spent the better part of 3 decades chasing a question everyone else considered unanswerable. Could you sequence DNA from ancient bone? The problem wasn't just technical, though the technical problems were formidable. Ancient DNA is not like modern DNA. Time degrades it. Heat destroys it. Bacteria colonize it, overwriting the original genetic text with microbial noise. A sample pulled from a Neanderthal fossil contains, on average, less than 1% endogenous DNA, meaning DNA that actually came from the Neanderthal itself. The rest is contamination, modern human genetic material from the researchers who handled the bones over generations, from microbes that moved in after death, from the soil itself. Separating the signal from the noise required technological precision that simply hadn't existed until the mid-2000s. The extraction process is like reconstructing a shredded document that has been left in a muddy field for 40,000 years, with 1,000 other documents shredded on top of it, and then being asked to identify which fragments belong to the original. From a sample no larger than a grain of table salt, drilled from the petrous portion of the temporal bone, the densest bone in the human skull, and the one most likely to preserve original genetic material, Pabo's team extracted, amplified, and sequenced ancient DNA. They analyzed specimens from 3 different Neanderthal individuals from Vindija Cave in Croatia, specimens that had sat in museum collections for decades. Working alongside Pabo were researchers including Richard Green of the University of California, Santa Cruz, and Johannes Krause of the Max Planck Institute. The results were published in the journal Science in May 2010. What they found didn't match the archaeological narrative, not even close. Based on the morphological evidence, the physical separation of skeletons, the distinct tool traditions, the apparent lack of hybrid fossils, geneticists had expected to find what decades of assumption had built: clean genetic separation. 2 distinct lineages, no meaningful overlap. Instead, the data showed something that stopped the field cold. Neanderthals had contributed genetic material to modern humans, not a trace, not background noise, a substantial founding contribution present in every non-African human alive today. Between 1 and 4% of the genome of every person of European, Asian, Indigenous American, and Oceanic ancestry carries Neanderthal DNA. Or so the first results suggested. This was a ghost population made visible, of vanished people whose biology had persisted even as their culture, their language, their identities, everything that made them Neanderthal rather than human, had dissolved into history. They had not been replaced, they had been absorbed. The 2 lineages had met, recognized each other across whatever cognitive and social distance separated them, and merged. The DNA was the proof. 40,000 years of silence broken by a sequencing machine in a German laboratory. The implications cascaded. Pabo's team had sequenced approximately 60% of the Neanderthal genome from those Vindija specimens. Later work, particularly a 2013 high-quality sequence published in Nature from a toe bone found in Denisova Cave in Siberia, would push coverage to over 50 times sequencing depth, the kind of genomic resolution that approaches what you can get from a living person. Comparing these ancient genomes to data from 1,000 modern human genomes from populations across 5 continents, the researchers could begin to map exactly where in our DNA the Neanderthal contribution lived and what it was doing there. Not from one event, multiple interbreeding episodes. The genetic signals in East Asian populations carry slightly more Neanderthal ancestry than those in European populations, suggesting that after an initial admixture event that affected the ancestors of all non-Africans as they first left the African continent, there were additional mixing events as humans moved further into Asia. The Neanderthal DNA was not randomly distributed through the modern human genome either. Certain regions carried it at much higher frequencies than random chance would predict. This was natural selection. These were genes that had been useful, genes that had been kept. Here's where it gets strange. The Neanderthal DNA most enriched in modern human genomes relates to immune function, skin biology, and fat metabolism. Researchers including Tony Capra at Vanderbilt University and Michael Dannemann at the Max Planck Institute analyzed Neanderthal-derived variants in modern humans using medical database records from the Biobank UK and other clinical resources. What they found was that Neanderthal genes are not passive passengers in our genome. They are active contributors to how we function. Neanderthal-derived variants affect skin color and hair texture, almost certainly reflecting adaptations to the lower UV radiation of higher latitude environments that Neanderthals had occupied for hundreds of thousands of years before modern humans arrived. Other Neanderthal variants relate to blood coagulation, influencing how our blood clots and potentially affecting disease susceptibility. Still others connect to immune response to toll-like receptors, the molecular sentinels that detect invading pathogens, variants that may have helped early modern humans survive diseases that had been circulating in Eurasia for millennia before our species arrived. not our genes, borrowed genes, genes that may have saved us. The genomic picture that emerged from comparing 299 ancient genomes spanning 45,000 years of European prehistory was one of staggering complexity, not a replacement event, not a clean handover, a merger, messy and prolonged and real, written into the cells of every person reading this whose ancestry traces back to Eurasia. 3 milligrams of extracted bone powder, 50,000 years of human history against a sample that fits comfortably between 2 fingernails. But the DNA dismantled more than just the replacement hypothesis. It also demolished the brutish caveman, because if Neanderthals and modern humans found each other compelling enough to have children together repeatedly across multiple continents and multiple tens of thousands of years, then the cognitive and social gap between the species cannot have been what the textbooks claimed. You do not produce viable offspring across a profound cognitive divide. You do not sustain interbreeding across millennia with a creature you regard as fundamentally other. The Neanderthals who met our ancestors were not the shambling subhumans of museum dioramas. They were people, different people, but people. We carry pieces of these individuals in our cells. Every person of non-African ancestry alive today is, in some small but measurable biological sense, part Neanderthal. The Neanderthal who contributed those variants to modern Eurasian populations was not some nameless primitive. He or she was a person with a genome sophisticated enough to confer immunological advantages that persisted for 40,000 years because they worked. That is not the story of a failed species. That is the story of a successful one, one that achieved a kind of immortality, not through culture or language or monument, but through the oldest medium of all, DNA. This affects how we breathe at altitude. This affects how our bodies respond to viral infection. This connects to traits as intimate as the texture of our skin and the architecture of our sleep cycles. Neanderthal-derived variants have been linked to chronotype, the internal clock that makes some of us larks and others owls, the daily rhythm of human existence encoded in part by people who watched the sun set over Ice Age European valleys 45,000 years ago. The shadow of their biology falls across our mornings. The same pattern of deep admixture has emerged elsewhere. From the Denisovans, a sister species identified entirely from a finger bone and a molar found in that same Siberian cave, modern Tibetan populations inherited a variant of the EPAS1 gene that allows efficient oxygen use at altitude. Without Denisovan DNA, the highlands of Tibet might have remained uninhabitable for our species. Populations in Papua New Guinea and across Melanesia carry up to 6% Denisovan ancestry. The ancient world was not a world of pure isolated lineages. It was a world of convergence, of contact, of biological exchange across every border we once imagined was fixed. Research by Joshua Akey at Princeton University and David Reich at Harvard Medical School has continued refining the picture, identifying additional interbreeding events, mapping the genomics landscape of ancient admixture with increasing precision. Reich's laboratory, which has published more ancient DNA studies than almost any other institution on Earth, described in 2018 the genome of a first-generation hybrid individual from Denisova Cave, a young woman whose mother was a Neanderthal and whose father was a Denisovan. Not a metaphor. An actual person, a daughter of 2 species living in a Siberian cave 90,000 years ago. The ancient world held multitudes we had not imagined, and somewhere, in a climate-controlled drawer in a German natural history museum, a fragment of bone no larger than a matchstick waits. It holds the genetic sequence of a Neanderthal whose descendants may still be walking this earth, whose biology shaped immune responses that have survived 400 centuries of selective pressure, whose existence we spent 150 years mischaracterizing as a dead end. It was not a dead end. It was a beginning, one chapter in a human story so tangled and ancient and resilient that no single fossil, no single genome, no single laboratory will ever read it whole. You can erase a name from stone, you cannot erase it from cells. The Neanderthals left no written record, no temple, no inscription that has survived the ice ages and the millennia, but they left something more durable than any of those things. They left themselves fragmented, diluted, altered by time and selection and 40,000 years of recombination, but present in a skin cell, present in an immune receptor, present in the quiet biological machinery that keeps you breathing through the night. The story of Neanderthal DNA is not finished. It is only beginning. Every year, new extraction techniques, longer sequencing reads, more sensitive computational tools for detecting archaic ancestry in modern genomes pull more signal from the ancient noise. We will identify new interbreeding events. We will map new Neanderthal variants to new biological functions. We will find, in all likelihood, that the merger was deeper and stranger and more consequential than even the current data suggests. Subscribe so you don't miss what comes next, because the next chapter will rewrite what we thought we had already rewritten.